So just as a recap of what we did last time, when it came to the digestive system, first thing that we went over, aside from the functions, and in the functions there are a lot of terms that you guys need to make sure you understand what they mean, like mastication, deglutition, peristalsis, segmentation. Can't think of any other right now, but anyway, all those terms which pertain to the functions of the digestive system, you should be familiar with. Then when it came to the anatomy, what we did is we divided the organs into those that are part of the GI tract and those that are accessory organs. You should definitely know both sets. And when it comes to the GI tract, you should be able to follow the food from the oral cavity all the way to the elimination of waste in the rectus, the large intestine. Obviously, also know which ones are the accessory organs. Now, this will be considered gross anatomy. When it came to histology or macroscopic anatomy, you should definitely go back and review the histological organization of the GI tract. And what we talked about were the layers of tissue, starting with the mucosa, which is the most superficial tissue, and it consisted of the mucosal epithelium, which was found starting in the stomach. Actually, let me go ahead and rephrase that one. Mucosal epithelium is in the esophagus, was stratified squamous epithelium. And then starting in the stomach, it became simple columnar and continued in simple columnar all the way to the end. Below the mucosal epithelium is a small layer of a connective tissue called the lamina propria. And then deep to the lamina propria was the muscularis mucosa. Okay. The next layer of tissue was the submucosa. And the submucosa consisted mainly of connective tissue. Depending on the location, there might be glands that secrete mucus, there might be Peyer's patches from the lymphatic system. And starting at the small intestine, also in the submucosa, there was going to be a plexus, which is the submucosal plexus. Which its job was to stimulate secretions. Okay. So again, the submucosa is essentially connective tissue. Then below the submucosa was the muscularis externa. With smooth muscle, two layers of muscles, except at the stomach there were three layers. And the other exception was esophagus. First 1/3 was skeletal muscle. And then the last layer was the adventitia or serosa, which again is connective tissue. Okay, so those are the histological layers of the GI tract, and you should know those. And you should know the exceptions and when tissue changes, and we have special structures in certain areas. We also talked about the mucosal epithelium, starting at the stomach, being made of these simple columnar cells, to this layer of simple columnar cells, which would, in uh, certain locations, dip below and create these pits or glands. And this would happen in both the stomach and the small intestine. And it is in these pits or glands where a lot of the interesting cells that secreted um, things that were needed for digestion were located. Okay, so that was part of the mucosal epithelium. So that takes us to the cells and their secretions, which are important for digestion. So in the stomach pits, we talked about parietal cells, chief cells, and enteroendocrine cells. Parietal cells secreted two things: intrinsic factor, which was needed to release and bind, well, actually to bind to vitamin B12, and then take it to the ileum where it will be absorbed, and hydrochloric acid. Chief cells secreted pepsinogen, which with the action of hydrochloric acid was converted into an enzyme called pepsin, which digested proteins. So this is all in the stomach. Enteroendocrine cells secreted gastrin, which is a hormone that would stimulate chief cells and parietal cells. Enteroendocrine cells also secreted histamines, which are not hormones. These are going to be paracrine secretions. And histamines stimulated parietal cells. As a matter of fact, the main stimulus of parietal cells and secretion of hydrochloric acid is histamines. So some of the medications to lessen the production of hydrochloric acid are actually antihistamines type medications. Okay, so those are the cells in the stomach. Then we talked about the cells in the small intestine. And actually what we did is we talked about the stages of the stomach secretion. And in a reference to the stages in gastric secretions, we talked about the cells in the small intestine that secreted hormones. Okay, so gastric secretion phases were the cephalic phase, which prepared the stomach for the arrival of food and initiated the secretion of gastrin, parasympathetic stimulation, and it was a short phase. This is the shortest of the phases. Then came the intestinal phase. Sorry, not the intestinal phase. The gastric phase. And this one started when food arrived at the stomach. And this will be full secretion and contractions. So this is the full work of the stomach. At this point, everything is being secreted: hydrochloric acid, gastrin, which in turn helps in the secretion of hydrochloric acid, histamine, which again helps in secretion of hydrochloric acid. Pepsin is being secreted. Intrinsic factor, So the full work of the stomach is taking place during the gastric phase. This is a very long phase, and it is stopped when a small bolus of chyme, of a small aliquot of chyme of the content, is sent to the small intestine, and then we have the intestinal phase. Okay, intestinal phase. So also the gastric phase, we have the parasympathetic activity. Intestinal phase, it stops the gastric phase due to the secretion of two small duodenal hormones. This happens indeed from the duodenum. It starts with the arrival of food in the duodenum, which is the first part of the small intestine. Low pH due to the arrival of the stomach chyme, undigested fats, lipids. That's what triggers the intestinal phase in the duodenum. The hormones secreted are going to be secretin and cholecystokinin, which the acronym is CCK. And these hormones are going to stop the gastric phase. And then we're going to see in a few minutes how secretin and CCK are also involved in the secretion of digestive enzymes. Okay. So there is a back and forth between the gastric phase and the intestinal phase until the contents of the stomach are all digested. Now, two other things that we haven't touched on are the effects of parasympathetic and sympathetic nervous system on the GI tract. And you should definitely know that. You should know that via the actions of acetylcholine, parasympathetic stimulation stimulates GI tract. Sympathetic, on the other hand, its neurotransmitters is norepinephrine, and it is going to inhibit the activities of the GI tract. All right, so that's a pretty good overview. I did not go over the stages of swallowing. You need to review that. But now we need to move on and look at what the accessory organs do and how they contribute to the functions of the GI tract. So as far as the liver, go back to lab and review the anatomy of the liver. A main thing here will be the ducts and the function of the liver. Let me go for the functions of the liver and then we'll pick up on the ducts. First of all, the cells that make up the liver are called hepatocytes. And these are quite amazing cells that do tremendous, tremendous work. They produce bile. Actually, the liver will also store bile, even though most of the bile storage is going to be in the gallbladder. It interconverts nutrients, so it may take glucose and make it into glycogen. It may store fatty acids. It may take proteins and make them into glucose. It detoxifies. It has phagocytic cells that roam around and phagocytize debris that may come from food. And it synthesizes blood components, synthesizes a clotting factor, synthesizes albumin, synthesizes complement, fibrinogen. I can't think of anything else. But I'm sure there's others that escape right now, not my mind, but tons of blood components are synthesized in the liver. All right, so those are the functions. Now, when it comes to the bile, bile is going to be produced by the hepatocytes in the liver, and then from there is going to be taken over to the gallbladder, which is the storage organ for bile. So bile will be collected by the right and left hepatic ducts of the liver, which are going to collect the bile and take it to the gallbladder via the cystic duct. Bile is going to be stored and modified in the gallbladder. And then when it is needed, cholecystokinin is going to cause the gallbladder to constrict and begin to expel the bile, which goes through the cystic duct into the common, oh, well, I forgot the little common hepatic duct, but that's okay. Into the bile duct. The bile duct is going to join with the main pancreatic duct and dump the bile into the duodenum by going through the hepatopancreatic ampulla. Okay. So that is the course that bile takes. So again, right and left hepatic ducts, which come together to make the common bile duct. The little duct forth from the gallbladder is called the cystic duct, and then we have the common bile duct that takes the bile to the duodenum of the small intestine. All right. The function of bile. Bile is alkaline. So remember that the acidic kind comes from the stomach into the duodenum, and the duodenum is not made to withstand acidity. So that acidity has to immediately be buffered, and that's part of the job of bile. Bile is going to dilute hydrochloric acid and is going to raise its pH, and that's going to create a better environment for digestive enzymes to work. Bile is going to emulsify lipids. Bile is very much like a soap. So it's going to separate lipids and make them available for digestive enzymes to break them down. It deals with the waste products and cholesterol. So it also carries cholesterol to the small intestine, and from there is going to be expelled through feces. Bile is released under neural control and hormonal control. So the neural control is under parasympathetic innervation. Hormonal control is under the stimulus of hormones, secretin and cholecystokinin. Secretin stimulates the production of bile. Cholecystokinin causes the gallbladder to constrict and release the bile. Okay. So this is just going over the production of bile, or the release of bile, I should say, not the production. So vagus nerve, which is a parasympathetic nerve, and that is going to innervate the gallbladder. The small intestine is going to secrete secretin and cholecystokinin, which are going to secretin stimulates the liver into producing bile and sending it to the gallbladder. Cholecystokinin is going to cause the actual release of the bile. Bile salts end up in circulation, and they also stimulate the production or the release of bile. All right, now when it comes to the other accessory organ, the pancreas, it is located with its head abutting against the duodenum and the tail against the spleen. This is an organ that has two functions. It has endocrine function and exocrine function. The endocrine function, we saw it in the endocrine system chapter, is the secretion of insulin and glucagon by beta cells and alpha cells in the pancreatic islets. Exocrine function involves the acini cells, which secrete pancreatic enzymes. Okay. Pancreatic secretions mix with bile, and they're secreted together in the hepatopancreatic ampulla, which is into the duodenum. Okay, so bile has a couple of the pancreatic secretions have a couple of components. They have ions and buffers, which again, like bicarbonate ions, which are going to act as buffers, trying to again buffer the acidity of the that arrives from the intestines into the duodenum. It also has digestive proteins, trypsin, or enzymes that digest proteins, trypsin, chymotrypsin, and carboxypeptidase. It has enzymes that digest carbohydrates, like pancreatic amylases. Has enzymes that digest lipids, pancreatic lipases. So what stimulates the secretion of secretin and cholecystokinin is the acidic chyme that arrives at the duodenum. Also, the presence of fatty acids and lipids will stimulate secretion of cholecystokinin, and obviously the stimulation by parasympathetic nervous system. So to properly digest, we need fatty acids, we need lipids, we need acids, we need an acidic production of acids in the stomach. And this again is just showing how the pancreas is stimulated. Vagus nerve stimulates the pancreas into secreting secretin and cholecystokinin, which are hormones that are going to travel through the blood and stimulate the secretion of the exocrine secretions of the pancreas. All right, so we're going to go into the anatomy of the large intestine. I guess here what you need to do is go back to the lab, to the practical, and review the anatomy of the large intestine. Let me see here. Oh yeah, just remember the ileum is the last part of the small intestine which connects to the large intestine. Cecum is the first part of the large intestine, and then it goes into the ascending colon, transverse colon, descending colon, sigmoid, rectus, anus. The types of cells we have in the large intestine will be absorptive cells, goblet cells, granular cells. So these cells are going to be absorbing water. Goblet cells secrete mucus. Granular cells also are going to have mucus secretions. Right, so most of the digestion occurs in the duodenum. Most of the absorption is in the duodenum and the jejunum. The stomach can absorb things like alcohols and aspirin. And then we're going to go over where each of the food groups are going to be digested. Carbohydrate digestion begins at the mouth thanks to the actions of salivary amylases. Then it stops in the stomach and it picks up again in the duodenum via pancreatic amylases and brush border enzymes. Remember, brush border enzymes are the enzymes secreted by the simple columnar epithelial cells that lined the small intestine. So those little lining epithelial cells do secrete digestive enzymes called brush border enzymes. Digestion of lipids, there's a little bit of it in the mouth due to the action of lingual lipases. However, and again, a little bit in the stomach, there's a little bit of gastric lipases. However, the bulk of the lipid digestion takes place in the duodenum, mostly by the actions of bile salt and pancreatic lipases. Proteins digestion begins in the stomach via the actions of pepsin, and then in the duodenum via pancreatic enzymes. All the nutrients that are being absorbed in the duodenum and the jejunum are going to be transported directly to the liver via the hepatic portal system. And this is a summary of the food groups and where they're digested. Part of the function of the GI tract is to absorb all the fluid that's been put out and ingested during a meal. So there is fluid that we use when we eat, we ingest when we eat, salivary glands secrete fluid, gastric secretions, and all these fluids accumulate, and in the small intestine, most of these fluids will be reabsorbed to be put back in the blood circulation. And then the large intestine also absorbs some fluids. but most of the absorption of water takes place in the small intestine. Okay, so that is basically it for the digestive system. Any questions? All right, I'm going to go ahead and stop these PowerPoint and start a new recording.